The stimulus is transmitted via activation of signaling proteins within the cytoplasm. This is followed by a transfer of the signal to the nucleus, where transcription factors specific for insulin will be activated to turn on gene transcription. The insulin gene is found on chromosome 11p15.5. A pre-mRNA is synthesized and splicing occurs to remove introns and join exons. The mature mRNA is transported out of the nucleus to the cytoplasm, where it can interact with the ribosomes on the endoplasmic reticulum.
The ribosomes receive mRNA from the nucleus, which is read and then turned into preproinsulin that goes from the ribosome to the endoplasmic reticulum.
As the preproinsulin is imported from the ribosome to the membranous sacs in the endoplasmic reticulum called cisternae, which isolates the internal lumen from the cytoplasm, the peptide signal at its end is removed, thus becoming proinsulin. Proinsulin is made of three parts: an A chain, a B chain, and a connecting peptide in the middle, called the C peptide. In the the C peptide is excised, and the A chain and B chain are connected by disulfide bonds. Insulin and the C peptide are then transferred to the Golgi bodies.
The Golgi apparatus receives the vesicles from the endoplasmic reticulum. These vesicles are carrying the proinsulin they enter through the cis phase. As the proinsulin is being transported through the Golgi towards the transphase, the proinsulin is processed in what is commonly referred to as insulin. Vesicles then carry the insulin from the transphase away from the Golgi. In addition, the Golgi apparatus processes digestive proteins received from the endoplasmic reticulum and packages them into vesicles that are more commonly known as lysosomes.
Membranes and proteins produced by the ER travel to the Golgi by way of vesicles.
Lysosomes receive enzymes from the Golgi apparatus that is used in the digestion of biological polymers like insulin. These enzymes, such as hydrolase, are synthesized in a rough endoplasmic reticular. The membrane-enclosed lysosomes function as garbage trucks, degrading proteins such as insulin when malfunctioned during protein synthesis. They also consume waste created from inside the cell when insulin is created. They undergo phagocytosis, in which particles are eaten by the enzymes in the lysosomes that happen by the fusion of the plasma membrane around the particle.
Production of insulin in beta cells, the plasma membrane regulates the release of insulin to the rest of the body.
Once insulin has been fixed to the correct form by the other organelles, it is not released until signaled by a change in the glucose level of the blood. Once the signal has been received, insulin exits the cell through exocytosis. Exocytosis is a form of active transport, so ATP is required. The vesicle holding the insulin merges with the plasma membrane, as both are composed of a phospholipid bilayer. The vesicle integrates with the plasma membrane and then opens the exterior side of the vesicle, so the plasma membrane is once continuous and the insulin is released into the space outside of the plasma membrane.
Mitochondria are the powerhouse of the cell, where cellular respiration takes place for ATP synthesis, a process vital to life. In ATP synthesis, energy is created for organisms to perform necessary daily activities. Mitochondria use chemiosmotic coupling to harness energy from high-energy electrons in stored carbohydrates. The enzyme ATP synthase then catalyzes the energy-requiring synthesis of ATP from ADP. When the energy is needed, a water molecule breaks a phosphate group away to release the energy. The energy is released by ATP state change into ADP, and the energy can then be used in other parts of the cell.
During replenishment, insulin granules have to be transported from more intracellular locations toward the exit sites on the plasma membrane. Microtubules and the motor proteins perform the long-distance transport of insulin granules. Subsequently, the motor protein accomplishes their transfer along short-actin filaments to the docking sites at the plasma membrane.